All right, pre-AP biology. We're going to continue today with our lecture notes on cell reproduction. We've covered a lot on this so far. We got a little bit more to cover, which I think is pretty important. So this is part 4. This is the note packet and the PowerPoint slide that goes along with that. Before I get started, I want to kind of review a little bit from last week, because there was a lot that we covered last week when it came to the cell cycle. So just re-looking at this slide here and this graphic that I gave you last week. Again, I want us to make sure we know, how a newly formed cell. So my cursor here is pointing at this newly formed cell right here. And that cell is going to go through a series of events in its life, right? It's going to go through G1, and if it doesn't, it'll go to G0. We talked about that last week, but it'll go through G1, S, and G2, which is what we call interphase. And there are certain things that go on in each one of those. And then that cell will enter the mitosis phase, where it goes through PMAT, right? Prophase, metaphase, anaphase, telophase. So I'm not going to go into the details of those, but look at my last lecture and you'll know that. And then it goes into cytokinesis, which is when the cell splits. All right, so once that cell divides, that means basically the life of that previous cell has ended, and we have the birth, I suppose, of two new cells. Okay, these are called daughter cells, but they're the results of one cell going through the cycle and resulting in two cells. Okay, then those two cells will go through the cycle, and then 4 cells, and on and on and on. Okay, so again, make sure that you review those steps. All right, there's a lot of vocabulary, a lot of stuff going on in this cycle that you have to know. Okay, now today we're going to talk about regulating the cycle, like the flow of the cycle. Okay, now the first thing here says is that not all cells move through the cycle at the same rate. Okay, so for example, like let's say that this newly formed cell here, let's say that this is a skin cell. Okay, skin cells live a couple of days basically, but I mean, they will go through this cycle and then they're done. Okay, so either they're going to divide or they're going to die, one of the two. But I mean, some cells, like skin cells, they go through the cycle fairly quickly. Some cells, like a brain cell, they go into G0 and they never go through the cycle. Okay. So, like, what influences whether or not a cell goes through the cycle or doesn't go through the cycle, or how fast it goes through the cycle? Basically, it's the DNA in the cell. Okay. What type of cell it is really kind of influences, how it's going to go through the cycle or how fast it's going to go through the cycle. Now, there's 2 other things besides the DNA that sort of influences cell division. One of them is called regulatory proteins, which I'll talk about. And the other one is called contact inhibition, which I'll get to after I talk about regulatory proteins. Now, regulatory proteins are basically chemicals that regulate the timing of the cycle. Think of it, I like to think of them as streetlights or traffic signals. So let's say, for example, I'm going to put myself up here. Let's say, for example, I don't know, let's say, oops, get my little electronic pen here. Oops, let's do it this way. There we go. Let's say that we've got a cell. Here's our cell here. And let me change colors. Let's get that to be a little bit. There we go. Let's use red. So there's our cell. Okay. So, this cycle here looks a little bit different from the last one I showed you, but at the end of cytokinesis, right, after the cell splits, we've got 2 new cells. So here's one of those cells, okay, and it's going to go through the cycle. It's going to go through interphase, which is the blue phases, and then mitosis, and then cytokinesis. But you'll notice, for example, say this cell goes through G1, okay, At the end of G1, there's a stoplight. It's like if you're driving a car on a road and you want to get from point A to point B, there's going to be some stop signs or some stoplights along the way. So this first stoplight, okay, this is a regulatory protein. It's a signal to the cell that basically says that you can't go on unless you've done all the things that are required in G1. Well, let's remember from yesterday or from the last lecture, right? During G1, the cell gets bigger and all of its organelles get copied. So if those two things don't happen, then the cell cannot move on into the next stages of the cycle. All right, but let's assume that the cell did what it's supposed to do, and now it's ready to move on to the next stages. So the regulatory protein will allow the cell to move on. So the cell moves into S, into G2, and it has to do whatever it has to do in those stages. And then there's another checkpoint. All right, so again, if the cell doesn't do the stuff that it does in the S phase, like replicate its DNA, for example, then it's not going to be able to move on. Same thing at the end of mitosis. There's a regulatory protein that regulates whether or not cells will move on. Now, if cells can't get past these check lights or these stop lights, then what will happen is your white blood cells, your immune system will destroy the cells. It'll just get rid of them, or the cells will just die. Okay, so that's kind of an important thing to remember because you probably do have some cells in your body that just aren't growing the right way. They're not going through the cycle the right way. And what happens to those cells if they don't do that? Well, your body has a way to get rid of those things. Okay. So again, just kind of make sure we're sort of understanding about what goes on with those regulatory proteins. Now, the other thing that could limit growth or can inhibit growth is something called contact inhibition. Contact inhibition is this idea that cells have a cohesiveness to them. Okay, cohesive. You might remember that term from previous lectures. It means that they stick together. So let's take a look at this picture here on the right. Now here we have a little petri dish, filled with cells. Let's say that they're skin cells that are grown in a lab or something. We'll talk about how that works a little bit more down the road. But let's say that in this dish, we have a layer of cells. Okay, and it's a thin, just a one-celled layer. They're all like nicely lined up next to each other. But your skin cells are sticky. They stick to each other. Let's say that we removed a chunk of those cells. We just cut them out and remove them. So now you'll notice that there's a gap in between the cells. Okay. Well, what cells do is they kind of understand. There's an actual like mechanism in the cell. It's all driven by your DNA that tells the cell that there's an area here that needs to be filled in. It's kind of like if you skin your elbow, right? If you skin your elbow, those cells are gone. There's a big gap there where cells used to be, and now they're no longer there. So what does your skin do? Your skin will grow new cells to fill in the gap, right? So what we would assume is that if we remove those cells that are lining the gap, they're going to reproduce and they're going to form more cells to fill it in. Now, normally it'll fill in nice and neatly. Sometimes they don't fill in perfectly. You might get a little scar there. That's what a scar is. It was basically when the cells that replaced the old ones didn't necessarily grow back exactly like they did before. So you get looks like some scar tissue there. Sometimes they'll divide abnormally, and that could lead to cancer. We'll talk about that more in a second. But this is contact inhibition. It's kind of like when you remove those cells, it's sort of a signal to other cells to divide until they fill in the gaps. Okay, so think about like a paper cut on your finger, or you skin your elbow. That's kind of what happens when those cells eventually grow back. All right. Trying to figure out my. Oh, let's just try that. That doesn't work either. Sorry, I'm having some technical difficulties with my cursor. Anyway, okay. So let's talk about abnormal cell division. Like, what happens if cells don't divide perfectly? Sometimes cells lose their abilities to regulate their own cycle. Okay. Like, for example, a blood cell. A blood cell goes through the cycle at a certain rate. There's certain regulatory proteins that regulate how the cell goes through it. But sometimes cells lose their ability to do that. These are called cancer cells. Okay. Now, cancer cells are basically cells that have lost their ability to regulate how quickly they go through the cycle. So it results in uncontrolled cell division. And the other thing that it results in is a loss of cohesion. So these cells are not only they're not only dividing at a much faster rate than normal, but they're also not sticking to each other like they normally would. Okay. Now, what this results in is something called a tumor. Now, there's two different types of tumors. Both of them, they're called benign and malignant. Both of them are the result of abnormal cell division. Okay. The difference between the two is that one of them, malignant, they have lost their cohesiveness, whereas benign tumors have not. Okay. If you go to the doctor and he tells you that you have a tumor, okay, automatically we think, oh my God, that's terrible, I've got cancer. Okay. Doesn't necessarily mean that. Okay. If the tumor is benign, that means those cells, even though they've divided uncontrollably, they're still in the same area. They haven't broken apart. Okay. Some good examples of benign tumors are warts. Have you ever heard of a wart before? A wart, I mean, sometimes people get them on their feet or their hands. Warts are just abnormal cell division, just cells that have divided abnormally, but they're all in the same area. So your doctor can just freeze it off, or they can cut them out. There's medications you can take to put on them that'll just make them fall off. It's not that big of a deal. But tumors that are malignant, those tumors have not only divided abnormally, but they've also lost their ability to stick together. So when that happens, those cells can enter the bloodstream. We call that metastasis. And when they enter the blood, they can travel to other areas of the body and they can spread the cancer that way. So malignant tumors are a little bit more serious because, for the most part, not only have they lost their cohesiveness, but they've also entered the bloodstream, again, which is called metastasis. Now, what can cause what can cause cells, to one day be normal, and then the next day they're not normal? They're dividing uncontrollably and they're cancerous. Well, one of those things is genetics. There are certain types of cancers that are um, genetic. Now, that doesn't mean that, okay, let's say your mom had breast cancer, or your dad had prostate cancer, or your grandpa had lung cancer. That doesn't necessarily mean that you're going to get cancer. Okay. But there is a genetic aspect to some types of cancer. If there's a family history of, I don't know, let's say prostate cancer, which is a cancer found in males. If there's a history of that in men, let's say your grandpa had it and your great grandpa had it or something, then maybe it's worth getting checked out. Especially when you get to be a certain age. But you can say that pretty much about most cancers. One of the things that doctors will ask you when you go get checkups and things like that is they'll say, is there a genetic history or a family history of any types of cancers in your family? That's a good thing for a doctor to know. All right. So, he might, advise you to get checked out every couple of years or something for colon cancer or a breast cancer screening or whatever it is, mainly because there's a family history in your family. Now, another thing that can cause it is carcinogens. Carcinogens are things in the environment, which could be things like chemicals in the environment. It could be ultraviolet radiation that could cause sunlight. But these are things that can get into your cells and disrupt your DNA. And if it disrupts your DNA, it's going to affect the way that your cells respond to things like regulatory proteins and contact inhibition, all that type of stuff. So, I mean, you probably can do a Google search, and it might be a good idea today to maybe type in the word carcinogen, or type in what are some carcinogens, whatever. There's thousands of them out there. All right, and it might be important for you to know some of those things. So, I don't know, like for example, on a package of cigarettes, there's a little warning, and the warning will say, hey, cigarettes contain carcinogens that are known to cause lung cancer. I mean, it says it right there on the package, and people still inhale cigarette smoke, even though there's known carcinogens in the product, right? So, I don't know. Take that for what it's worth. But we don't want to put those things into our body. All right. Now, I do want to just kind of show you this last little graphic here about metastasis that I talked earlier. So let's say, for example, um, right here in these cells in green. Let's get my little highlighter up and going again. Darn it. Here we go. So let's say here, these cells in green. Let's say this is a tumor. Okay. And those cells have divided uncontrollably, and they've lost their cohesiveness. So what you're seeing here is kind of a little rogue cell that kind of broke off from the tumor, and now it's entering the blood. Okay, this is a blood vessel up here. And if it enters the blood, then it can go anywhere. People might get diagnosed with, let's say, a lung tumor, let's say a malignant, metastatic lung tumor, but that tumor can spread to their brain. It could spread to their kidneys. It could spread to other areas. That's kind of the dangerous thing about cancer is you don't really know where it's going to spread. Okay, we do know that there are some treatments and some therapies for certain types of cancers. You've probably heard of things like radiation therapy. You've probably heard of things like chemotherapy. And what they're trying to do is they're trying to not only kill those cancer cells, either using radiation or chemicals, but they're trying to, hopefully stop them from getting into the bloodstream. But if they're already there, you just don't know where they're going to go, and that's what makes it really kind of a tricky disease to treat. All right, guys, that's it for me for today. If you want to rewatch the video, feel free to do that. I will talk to you soon. Take care.